Hello, everyone, and welcome to this edition of the CINECA webinar series, where Kaur Alaso will present modular and reproducible workflows for federated molecular QTL analysis. My name is Marta Llorell Linares, and I'm part of the CINECA project on behalf of MBLBI. I will be hosting this webinar today. I would like to make you aware that this webinar is being recorded, and the recording will be available on the CINECA website and CINECA YouTube channel. We have reserved some time at the end of today's sessions for question and answers from the audience. So if you have any questions, please write them in the question and answer function on Zoom, as you can see here on the slide. I will briefly introduce the CINECA project to you. CINECA stands for Common Infrastructure for National Cohorts in Europe, Canada, and Africa. And it's a 4-year project funded by the European Union's Horizon 2020 program and the Canadian Institute of Health Research. CINECA's vision is to accelerate disease research and improve health by facilitating transcontinental human data exchange. And to achieve this, CINECA is working on a series of challenges. The first one is federated data discovery. So how, as a researcher, you can find cohort data of interest to you by genotype or phenotype, for example. And then once you find the data, you will need access to it. So CINECA is working on an interoperable authentication and authorization infrastructure. And once you get access to the data, if you want to ask research questions across cohorts, there will be a need for harmonized cohort metadata. The ultimate aim of CINECA is to enable federated data analysis in research and healthcare settings. So that's why CINECA works with a series of demonstrators across cohorts in both research and clinical settings. And on top of all of this, there's a need for a transnational ELSI framework to address ethical, legal, and societal issues that are associated with all this work. CINECA also has an outreach and training program to raise awareness and increase capacity in all these areas. So if you want to stay informed about what CINECA does, follow us on Twitter at CINECA Project or visit our website. Today, our presenter is Kaur Alaso, a lecturer of bioinformatics at the University of Tartu. His research is focused on genetic analysis of diverse molecular traits and building data resources to facilitate secondary use of molecular QTL datasets. Kaur is also a co-lead of the eQTL catalog project. And at this point, I will hand over to him.
Thank you. It's my pleasure to present in today's webinar. So I'll talk about modular and reproducible workflows for federated molecular QTL analysis. So many of you have probably heard about genome-wide association studies. And if you haven't, in very simple terms, genome-wide association study takes individuals who have some kind of a disease or a trait called cases, and then they take a control group, and then they test all genetic variants genome-wide for association with that particular disease. So basically, they look for if certain genetic variants are more common among the carriers of the disease compared to controls. And a typical endpoint of a genome-wide association study is a Manhattan plot, something like this, where on the x-axis, you've got genetic variants on chromosomes 1 to 22 at X, if you're lucky. And then on the y-axis, you've got the association p-value, showing how strongly a particular variant is associated with the disease or trait of interest. And what you can see here is that there's lots of and there's the typical threshold for statistical significance is here around 10 to the minus 5 times 10 to the minus 8. And what you see, there are lots of regions that are significantly associated, and then these regions have gene names linked to those. But any of you who is more familiar with genome-wide association studies would know that most of these gene names here are fiction. And they're fiction because most genetic variants in genome-wide association studies are non-coding. So there's only less than 10% of the variants in a typical GWAS are in the coding sequence of a gene, and then they disrupt, they change the mRNA sequence and they disrupt the protein. So they lead to a non-functional protein. And most, typically over 90% of the variants identified in a GWAS are located in regulatory elements, and they act by increasing or decreasing the expression level of a gene. So you get more or less RNA, and then you get more or less protein. So how do we link these regulatory variants identified in a GWAS study to their target genes, the causal genes that lead to disease, for example? So the simplest one is you would assign each variant to the gene closest to it, and that's typically done in the plots, but it's not always accurate. And then the second strategy is that you check for association with gene or protein expression levels. So you look if the variant that's associated with disease or a trait is also associated with expression level of some gene or protein in a specific cell type or tissue. And that's what we'll focus on today in this webinar. And just to highlight that there are alternative strategies as well, which we are not going to talk about today. So here is one example where we have a GWAS signal zoomed in for, in this case, for lymphocyte count on the top panel. Again, we have genetic variants on this particular region on chromosome 2 on the x-axis, and then we have the association log -10 p-values on the y-axis. And so we see a very strong signal in this region that's associated with lymphocyte count. And what we find is that the same region is also associated with the expression level of RBMS1 gene expression level in one specific cell type. And the signals look very similar, so we can do a, if you want, we can statistically test if they're likely to share a causal variant. In this case, they are. So the colocalization posterior probability is very close to 1. One thing we've noticed, though, is that these genetic effects on gene expression are often highly context specific. So they're often highly tissue or cell type specific. So here, what we did is we picked the same variant associated with lymphocyte count and looked at it across many different cell types and tissues in the eQTL catalog. And what we see is that we only see significant association with the expression level of RBMS1 in lymphocyte subtypes, especially in CD4 positive T cells. And if you look at other, for example, brain tissues or other tissues in the body, we see no association. So we only see this in a particular subtype of T cells. And so the caveat of this is that to be able to find this association with gene expression, we need very large data sets from very diverse cell types and tissues, and we need to process all of them in the same way. So that's what we've been doing in the eQTL catalog project. So the current solution in the eQTL catalog is that we bring all of the data to a single location and then process the data locally. So we start by identifying where the data are located. So they are typically in EGA or dbGaP or other databases, either controlled access or open access. Most of them are controlled access. And so we identify the data sets, we apply for access to them, and then we get the raw data. And then we have built uniform workflows to process these data sets. So for genotype data sets, so information about genetic variants, we take the raw data, perform quality control, and then impute it to the latest version of the 1000 genomes reference panel. And then for the RNA sequencing data, again, we take the raw data, we process this to quantify gene expression and the number of other traits, including exons and transcripts. We get a raw gene expression matrix, and then we perform additional quality control, and then we normalize the data, and then we perform association testing, after which we get summary statistics that we can publicly release on the FTP server and the API. And so all of these results are publicly available. So here's our publication, and here's the website for the eQTL catalog. So currently, eQTL catalog, so the latest release 5, contains 25 RNA sequencing studies and 6 microarray studies, which means we have uniformly processed around 30,000 samples from more than 7,000 donors. We cover 74 distinct cell types and tissues, and as a result, we have data from 125 data sets. So one of these studies is GTEx, and GTEx contains 49 tissues. So we would have from the GTEx study would be what we would call, GTEx is one study containing 49 data sets. But we have many additional ones as well. So what are the challenges with this existing approach? So the first challenge is that setting up data access agreements is really time consuming. So we've spent a good part of the past 3 years applying for access, writing applications, applying for access, getting denied and asked for clarification, and finally getting access to these data sets. And the second challenge is moving large data sets is often either impractical or completely impossible. So for example, we did download the full GTEx dataset from dbGaP, and it took us around 2.5 months to download all of the around 100 terabytes of raw RNA sequencing data. Whereas if we had analyzed it in place, we would have completed the whole analysis in maybe a week or two. And the second limitation is that there are a number of data sets that have, for example, they might have data locality restrictions. So you're not allowed to move the data outside of a specific country, or you might not be allowed to move the data outside even a specific data center. And then the only option is to analyze the data where it is located, rather than download the full data set to your home institution. So how do we move our data analysis code to the data? So how do we go from this central database model that we also use in the eQTL catalog right now, predominantly, where we take data from different locations, move it to a single server or single compute infrastructure, and then perform all the analysis there? How do we go from this model to a federated approach, where the data is located in many different places in the world, and we send our analysis code or our data analysis workflows to these places where the data is located, and then get the results back? So how do we do that? Currently there are for these type of workflows that we are working with, which are very intensive processing of raw data, there are basically two practical approaches to perform federated analysis. One is what I would call manual federation. This is actually how most genome-wide association study meta-analysis are performed. So what you do is the researcher sends their kind of analysis code to a local analyst who has access to the data. The local analyst will then perform the analysis, validates the results, and then sends them back to the researcher. So the researcher themselves actually will never have access to the individual level data. And then researcher combines the results from multiple locations in a meta-analysis framework once he or she has received the results from different locations. And the second alternative is are these trusted research environments, or TREs. And so in this model, researcher first obtains legal permission to view individual level data. So it means you still have to actually go through all of the data access agreements, because you will get access to you will see the individual level data, and so you need legal permission to be able to access it. The The researcher will then perform the analysis within the secure environment of the TRE. And once the analysis is complete, the researcher asks for permission to export the results. And typically, the results are then reviewed by somebody who controls the TRE to make sure that only the data that's allowed to leave the TRE leaves the TRE and nothing more. And then researcher continues with the same meta-analysis framework as in the case of manual federation. And this is becoming more and more popular. So currently most large biobanks or large human cohorts are either using this TRE model or are moving towards that model. So here are some examples. UK Biobank famously recently, subset of UK Biobank data is only available in their TRE. For example, their whole genome sequencing and exome sequencing data can only be accessed and analyzed in the TRE. Genomics England uses a TRE where the data can be accessed and analyzed. The FinGen project in Finland uses a similar environment, and recently established the Danish National Genome Center is also following this model, where they have a trusted research environment where then qualified researchers are able to access the data. So what are the pros and cons of these solutions? So even though manual federation seems like an old-fashioned way of doing things, it does actually have some advantages. Mainly, it means that the researcher who kind of leads the study does not need access to individual level data. So if there's a local analyst who can run the analysis and who has all of the permissions to do that, then this can actually, sometimes this can significantly save time by reducing the amount of time required for to obtain access to the data. But there are also cons to this. Namely, researcher has no control over how the code is being executed. So if there are any issues, they're very hard to troubleshoot because you're kind of blind and relying on the local analyst to tell you what the problem is and then trying to figure out the solution. And perhaps even more importantly, manual federation can only take place in kind of this ad hoc setting where you need every time you want to do something, you need to set up, you need to manually set up agreements with the data owners to be able to run the analysis and figure out how to compensate them for their time and computational resources. And often actually that's the main bottleneck, is the data is there, but it's not possible to kind of come up with an agreement to allow researchers to use the data. So trusted research environments overcome some of these limitations. Namely, the researcher has complete control over the analysis code. So if there are any issues, they are very easy to fix, and it's the right person who really wants to analyze the data who is then responsible for fixing the issues. So that's a plus. The other thing is that all of the bureaucracy involving data access, usage tracking, billing, etc., they're handled by the TRE. So typically there is a well-defined process of how a researcher would gain access to the data and how the researcher would then pay for processing the data. And that means that it's usually, if the researcher is qualified, it is possible to get access and analyze the data. But as I said before, it does require applying for access, which can be a significant amount of time. My recent experience with UK Biobank was around 2.5 months to get access, which is actually, I would say, probably it's about average time it takes to get access to the data set. Is definitely a few months. And the cons of TREs is that setting up a TRE requires a relatively large upfront investment from the data owners, and so far it has been typically done only by very large biobanks. And the other con is that you still need manual review of the results before they can be exported. So even though it's an approach to do federated analysis, there's still a manual step involved in reviewing what gets exported. And I don't really see a very good way of avoiding that manual review if there are requirements that the individual level data cannot leave the theory. So this is a summary from one of the theory providers, LifeBit, where they summarize how this manual review works. So you can run analysis, you can create results, but they're typically locked in the platform. Any movement of data into and out of the data platform goes through, in this case, they call it an airlock system, whereby the data movement is subject to review by an authorized team. It is essentially an independent validation that the specific data is authorized to move. And I think this is a kind of one of the bottlenecks that it's hard to see that it's going to be solved anytime soon for the general case of federated analysis. There's always going to be some manual review of the results that leave the analysis environment. But regardless of this, how can CINECA reduce the friction in existing federated analysis frameworks, both the manual but also the TRE environments? And so in the CINECA project, we have multiple work packages. We work on the federated discovery, authorization and access, harmonized metadata harmonization, and then federated analysis. And actually, for the federated discovery, there has been a webinar last year that you can find on the CINECA website, and also following this link after the presentation when you get the slides. For the cohort level method harmonization, there's also has recently been a webinar that you can see. And today I'll mainly focus on the federated analysis interoperability. So how to make it easier to move analysis code between different data infrastructures or these TREs. So how do we move around data analysis code with minimal friction? So I think when we started on these ideas, maybe like 10 years ago, it was typical to make your data analysis scripts available in repositories such as GitHub. But then we soon realized that it's nice to have these scripts, but it's almost impossible to reuse them because it's very hard to install all of the dependencies. So specific versions of specific software packages that you need to be able to run these scripts. So then there was an idea that you could put all of these dependencies into a single Docker Singularity image or a virtual machine, and then you could move that around. But that also, actually building these very large images can easily become complicated, and it really doesn't solve the issue where you need to, for example, run lots of analysis in parallel. So then what I think what the field is moving towards is that you can make a separate image, a separate container image for each tool. But then you have lots of scripts. Each script has its own image, and then you need to manually orchestrate all of these scripts and the containers, and it's very easy to make mistakes. And in practice, it's very difficult to do. And in the settings, like I showed, the eQTL setting, we really need to be able to parallelize our analysis. For example, if we have these 30,000 RNA sequencing samples, we can't analyze them in a sequence. We need to be able to rapidly run them in parallel on a compute cluster or a local or a public or a private cloud. And so the solution that we found that works quite well in practice is that you use a workflow engine that is able to execute complex workflows in parallel on multiple computational infrastructures. So that means public clouds such as Google and Amazon, but also private HPC environments that, for example, use Slurm as a queuing system. And these workflow engines would then also keep track of dependencies. So there are a number of competing solutions out there. For example, the Broad Institute has their own system called Whittle and Cromwell. What we are using in CINECA Work Package 4 that I'm talking about right now is the Nextflow language and the Nextflow workflow system, which also I think it's hard. It's not possible to say that it's kind of the standard in the field, but it's definitely widely adopted by many people in the field, and it has very good support for different computational infrastructures. So how do we approach this eQTL analysis problem then? So what we've come up with trial and error is that it's instead of having a single workflow that does everything from the beginning to the end, it's much better to have kind of modular sub workflows or modular sub workflows that do one specific task really well. So we have, for example, in the eQTL catalog, we've got 4 different workflows. So we got gen impute for genotype imputation. By the way, these names here are GitHub user and then the repository names. All of these things are publicly available on GitHub. So we have workflow for genotype imputation. We have workflow for RNA sequencing quantification. We have workflow for quality control and normalization, and then we have a final workflow for QTL analysis and fine mapping. So what do these workflows look like? This is a kind of a summary level of, for example, the gen impute workflow, where we take the raw genotype data in Blink format, and then we take a reference panel and some other inputs, and then we have multiple different steps that depend on each other. We kind of make sure that the genotype they use is the correct reference genome version. We then make sure that genotypes align with the reference panel, do filtering, phasing, imputation, and then kind of get the final results in the end. And we do it separately, for example, for chromosomes 1 to 22, and then the X chromosome, which need to be handled separately in imputation. And we have similar workflow for QTL analysis. We take normalized trait matrix, gene expression matrix, or some other trait matrix, and metadata and genotypes, and then we again do multiple different steps to get final results. And some of these things happen in linear, and then some of these end steps we can really parallelize them across large number of compute nodes. So what is the value of this modular architecture? So the main thing is that it's really easy to reuse these workflows in, for example, in a different domain. So for example, if instead of gene expression, we want to analyze chromatin accessibility, we can follow basically the same model where we get the raw data from different repositories. We reuse the genotype imputation part because that stays the same. We take a different workflow to quantify chromatin accessibility and normalize it. There's a public workflow available from the NF-core repository that you can use, and we have used this. And then you take the output from this NF-core workflow and the gen impute, and then reuse the QTL map workflow to get the association summary statistics. So you can kind of take these modular bits and combine them in different ways without having to write a lot of new code. Another application is so in the eQTL catalog, we've done something that's called cis-eQTL analysis, where we only look at variants that are near the genes that we study. But we've now more recently moved to trans-eQTL analysis, where we test all variants against all genes. So basically what we're doing is, in each data set, we're doing around 12 to 15,000 GWAS studies for each gene separately, and then we're meta-analyzing those. And so what we do here is, again, we can reuse the genotype imputation, we can use gene expression quantification normalization, but we have rewritten the association testing and the meta-analysis workflows to better scale the amount of data that we have. So I talked about I mentioned federated QTL analysis. So do we actually do any federated analysis? Yes, we do. So we have an ongoing project with the Helmholtz Center in Munich to process eQTL data from stimulated monocytes. So that's a data set that is local to Helmholtz, and it's basically very difficult to move it out of Helmholtz. It's much easier for us to analyze it locally there. We also have another project where we're running trans-eQTL meta-analysis in lymphoblastoid cell lines across 7 cohorts. And actually 6 of them, we have the data in Tartu, but one of them is the Colos cohort. The data is located at the University of Lausanne. And again, we can't move the data out of their environment, but we can move our analysis workflow there and run the analysis. And we've done that, and it worked really well. We now have the results that we're looking at. And we also have another collaboration together with the University Medical Center Groningen, where we are looking into federated trans-eQTL analysis in whole blood across 20 plus different cohorts. So a bit of what do I think? What do I think that might happen or might be useful for the future? I think I talked to you about this manual federation, and I talked to you about these trusted research environments. And I think one thing that can be helpful in the future are these trusted research environments that use synthetic data. And so the idea here is that instead of researcher applying for access, the researcher can immediately get access to a theory, but the theory doesn't contain the real data, but it contains synthetic data that's modeled on the real data. And so that means that the researcher can develop the analysis code and workflow and can test that it works, but won't really have access to the sensitive individual level data. And then once the researcher is happy with the workflow and is happy that everything works, he contacts the data owners so that the workflow is then transferred to a private workspace in that same TRE, where a local analyst can then run that workflow on actual real data. And the advantage here is that this should really kind of make things much easier to troubleshoot, because if the synthetic data closely matches the real data, then there should be much less issues of workflow failing because the data is not as expected. And then again, once the results are reviewed, they are exported and given to the researcher, and then they can be combined in the same meta-analysis framework. And at CINECA, we've been working on quite a number of generating quite a number of synthetic data sets. Can be used either in the applications, or they can be kind of modeled on to build synthetic data sets for specific cohorts to enable this type of federated analysis. Just wanted to put a link here that for the eQTL workflows, we do have tutorials available, and the workflows are available, so you can kind of access them here. And finally, I'd like to thank all of the people who have contributed to the work that we described today. So from University of Tartu, Nurlan and Ralph have been working on the QTL map and GenImpute workflows. From Helmholtz, Will and Anshika have been working on have helped us set up the federated monocyte eQTL analysis. And then Anneke Primmer and Sven Bergman contributed to the LCL lymphoblastic cell line meta-analysis, and Harmian and Robert from Groningen have contributed to the whole blood meta-analysis. And then, of course, all of the other members of the CINECA work package and I probably missed some important names here. And with that, I'd like to acknowledge funding for the project, and I'm now happy to take any questions that you might have.
Thank you very much for that presentation, Kaur. We have already one question that says how to get access to the platform. Is it freely available?
I'm assuming this is so I'm not really sure which platform the question is about. If the question is about the eQTL catalog, then that's freely available. All of the workflows used by eQTL catalog are also freely available. If it's a question about these trusted research environments, then these are usually set up by individual cohorts, and they have their own kind of criteria for accessing those. And they're typically not freely available. So UK Biobank charges you, I think, £9,000 so that you could access their TRE, and then they will, well, that depends on what type of data you want, and then they will also charge you for compute.
Okay, thank you. Yeah, I'll add in a moment the links to the eQTL and your GitHub catalog. But I also have a couple of questions. So you show these workflows you have for the different parts of the analysis and so on. You show that it can be for different analysis. I mean, you can use different workflows. So are you working on some other workflows? And if so, which type of workflows are you adding to this list of workflows?
So one of the things, that's a very good question. One of the things that we're working on right now is the genotype, for example, the genotype imputation workflow that we have right now only work with so the genotype imputation workflow that we have right now, GenImpute, only works with genotyping microarray data, and we're now working on another workflow that also supports low coverage genome sequencing. So that's something that's going to come out soon.
Okay, and is there a way that people can ask you for some workflows they are interested in, and then maybe you can work on them or so? You know, if someone wanted to analyze data from some other source or so, could they say, oh, are you thinking about doing this workflow? This would be very interesting for us.
I think people definitely contact me over email or Twitter, and I can often, actually, there are existing workflows. For example, NF-Core has a very large collection of existing workflows, and so we basically, our go-to is NF-Core, and if the workflow is not there, only then we will look into developing our own. We usually don't do it on demand, but if there's a convincing case that this is something that we're not doing and we should be doing, then I'm always open to suggestions.
Okay, thank you very much. So there are a few more questions on the list now. So what role do GA4GH standards play in running the workflows?
That's a tricky one. The short answer is short but maybe not very useful answer is that Nextflow task, for example, has a support for task execution service, which is a GA4GH standard, but it's not clear that it is very practical right now. So we have tested it, it does work, but you would have to have this service running in a Kubernetes cluster, and then you could kind of run Nextflow directly there anyway. So it's but it does in principle support that, but you need a shared file system, which makes it kind of tricky to run. So the other standard, which is workflow execution system, and it's at this stage it's not clear how, for example, Nextflow, or even actually the other workflows, how they would play with the workflow execution service. So it seems like Nextflow itself would be a workflow execution service, but it's not compliant with the standard, and it's a bit of a kind of there's a bit of an issue with what is kind of in the standard and what you can actually use for practical and large-scale analysis right now, and I think these two things do not always agree, at least at this stage. But I think that there's maybe a better. I think the tools registry side, where for example, you have containers containing software, I think that's also that should be relatively compliant with the GA4GH standards.
Okay, thank you. And then there's the next question is: are these TREs provided by companies? Is LifeBeat the only player?
They are typically provided by companies. LifeBit is not the only one. UK Biobank actually has, I think it's 7 bridges, and there are a number of other ones that provide these. They are typically commercial entities that develop these TREs. Yeah.
Okay. So the next question is, well, it starts with, thank you for a great talk. Then how would your workflows perform on non-European data?
That's an excellent question. I think for a gene expression quantification, I don't see any issues of like which population data is from. For genotype imputation, we use the 1,000 genomes reference panel, and that means that actually if there is a genotyping data set from a population that's not well represented in 1,000 genomes, then the workflow, at least in the kind of the default setting of the workflow, would not work very well. So if you have a very specific population in mind that's not represented well by 1,000 genomes, you probably want to have your own reference panel. And then actually these possibly use GenImpute with your own reference panel from that particular population. The software is standard. The software works with any population. It's a matter of, is the reference data set good enough? And QTL mapping, it runs a simple linear regression. So if there's a standard caveat that if there's lots of population structure, again, you have to be careful and maybe look for other models.
Thank you. Then outside Nextflow and WDL, how feasible is it to work with other workflow languages in such a federated environment?
I think the practical answer is you should look at what's supported by the TREs that you want to use to run your analysis. So at the moment, I think Nextflow and Middle are kind of best supported by the different TREs. So UK Biobank TRE, I think, supports Nextflow. I haven't tried it, but I think it does. There's an example on their website, and LifeBit, for example, supports Nextflow workflows. If it's something that's kind of if it's not directly supported, then it might be a bit more tricky to get things running. Although I think it's probably still possible, but it might be actually easier to use something that's well supported.
Okay, thank you. And then regarding your idea of using workflow first in synthetic data sets, does that mean each cohort should have its own synthetic data set, or a general synthetic data set could be used for different cohorts?
So the answer is yes and no, in a sense that so I think each cohort should have their own synthetic data set that kind of, especially if there are like specific phenotypes of interest, I think they should probably generate that themselves. But there could be aspects of the synthetic data set that could be borrowed from a common shared one. For example, there are large cohorts that have genotype data, and they might reuse, like if there's a very large synthetic data set of genotypes, then that could be reused by multiple different projects instead of generating that. So you could take kind of bits of kind of a shared data set and then add your cohort specific information. I think that will make things easier for everybody. And there are certain, for example, generating synthetic genotype data is actually quite tricky and computationally quite expensive. So if there's a reference data set that people could use, a very large one, which actually CINECA 4 partners are working on 500,000 individual synthetic genotypes, then that could be easily reused by different biobanks without thinking about how to generate this without leaking information.
Thank you. Then we have one about training. So thank you for your great presentation. Is there any plans for upcoming workshop or training soon? So Kaur, you can answer from your side, and I can have a quick answer from the CINECA or package 6. Let's say.
From the eQTL workflow, the molecular workflows, we don't have any immediate training, kind of hands-on training planned. We are working on the documentation though, and then the kind of the training material. And kind of improving the tutorial, but we don't have immediate plans for hands-on training or online training right now.
Okay, thank you. So CINECA is working on some plans, and one of them is also producing some training material. We will run some more webinars in the next months, but there's no defined date yet. And we will probably also run a couple of workshops next year, but again, it's not completely defined when or where. But if you keep an eye on our website, it should come up there. So, but we expect maybe to have some more things in February, March there, that time more or less. So we have one more question. Could also elaborate on the underlying frameworks that facilitate for the federated access to the platform? And lastly, technically, how challenging did you find it setting up the federated access?
So the simple answer is that for the three federated use cases that we are doing right now, we are using this kind of manual federated framework. So we have the workflow, and then actually there's somebody local to that HPC environment that kind of runs the workflow, which is in these small dataset settings that seems to be actually the most practical solution. Think about the technologies I think where the technologies are relevant are big biobanks, such as, for example, UK Biobank and Genomics England, and I think both of them they're using, one of them is using LifeBit, the other one is using, I think, 7bridges as the TRE environment, and both of them are running on AWS, so Amazon public cloud. And I think technically, I think they're commercial providers. So if you pay them well, it's not hard to set it up. It has to be funded from somewhere. So I think, for example, I think UK Biobank has certain pharma funding involved as well to help set it up, and FinGen has pharma funding involved to set it up. So it's definitely not for free, and I don't know any good kind of completely open source environments that would do this well right now.
Okay, thank you. So we have a couple more minutes if someone wants to still ask a question. I just wanted to remind you that, yes, this video will be available on the CINECA website and in our YouTube channel tomorrow or Monday. I mean, we'll process it as soon as possible. And then you can access both the video and the slides. So you can take a look at what we've been talking about today. And you can access also the links that are on the slides, and you can visit all the other webinars on the CINECA website. So if there's nothing else, thank you very much, Kaur, for your presentation. And thank you very much, everybody, for attending and for your interesting questions.
Yeah, thank you for the discussion. I really enjoyed it.